In Chapter 8, we delve into the importance of bacteria in forming and assisting our development of genetic techniques and our exploration of the genome of both humans and other species. So, bacteria have been critical in this, and we're going to go through the ways in which bacteria are able to contribute to science through their interesting mechanisms of genetic exchange and the feasibility of manipulation of their genomes. So, the first thing you need to know is that bacteria are a fantastic system for studying in the laboratory, in part because their populations grow so rapidly. If we were to study elephants in the lab, it takes 2 years just for the birth of a single elephant. It's not feasible to study large numbers of individuals and come to broad scale conclusions based on observations of a species that reproduces so slowly. Here you can see if we start with a very small population of bacteria, within a few hours that population can increase so dramatically in number through this exponential growth or log phase of population growth that we can produce millions and millions and millions of individual bacteria in a very short period of time. Okay? This is critical to some of the things that we use bacteria for, and some of that is going to be touched on at the very end of the course as we discuss how we are able to harness bacteria to produce things for us, such as insulin. Okay? All right, so within the lab, bacteria can be grown in a number of ways, but primarily they are grown in either liquid broth or on solid plates. So the material that's in that liquid or in that solid gel-like material is full of the nutrients that bacteria require. Now, bacteria produce most of the necessary organic compounds that they utilize, such as amino acids, okay? And those bacteria that are capable, they have this wild type of being capable of producing all of these necessary organic compounds. They are called prototrophs, and prototrophs can be grown on minimal media, meaning it provides the minimal amount of nutrients, minimal diversity of nutrients for the growth of bacteria. It gives them the bare bones, basic nutrition that they require to grow and survive. Okay. Now, on occasion, some bacteria will lose the ability to synthesize these specific compounds. Not all of them. They might lose the ability to produce one or two or more. But any of those bacteria that are incapable of producing a necessary organic compound are called auxotrophs. And as you might imagine, auxotrophs are incapable of surviving unless their diet, if you will, their media, is supplemented with that particular organic compound that they are not able to produce. So, an example in humans might be vitamin D, right? You produce vitamin D when you are exposed to sunlight. If you are locked in a dungeon, never exposed to any sunlight, you are not going to produce sufficient amounts of vitamin D, and your diet would have to be supplemented with vitamin D. Okay, it's the same principle here with these bacteria. So it's important that you understand the difference between these prototrophs and auxotrophs, and understand the difference between minimal media and enriched media. Enriched media being media that have been supplemented with whatever it is the bacteria need that they should be able to make on their own. Just to give you an idea of what these two different types of cultures look like, on the left we see examples of bacteria that are being grown in liquid media, and on the right is the solid media. On the left, we've got two different test tubes with kind of a yellowish liquid in it. This is what bacterial media looks like. And before you put bacteria in it, it appears quite clear, almost like apple juice. But over time, if you put a single bacterium into that clear media on the left and you let it sit overnight, by the time you come back the next day, the media will appear cloudy. It's cloudy because the population of bacteria has grown from one to millions. And so it is all the little bacterial cells floating in that liquid that are making it cloudy or opaque. Okay, so this might be something that you've seen before if you have, let's say, a fish tank that gets dirty or something that goes bad in the fridge that's got a bacterial culture growing in it. It goes from being clear to cloudy. On the right, we have a bacterial plate. So this is a small plastic dish that's about 3 or 4 inches across, and media is poured into this. It's basically the same media that's used in the liquid with a solidifying agent added to it. So you heat this media up, cook it all together, and then pour it into these plates, and it solidifies when it cools to room temperature. Okay, so I encourage you to think of this media as being just a solid form of the liquid with something added to it. It's basically like Jell-O. Okay, so you could think of the right being like Jell-O and the left side being kind of like Kool-Aid, right? They look the same, but one of them is solidified and the other maintains its liquid form. Okay, both of these are very useful in a laboratory setting, and they're used. Their use is dependent on what actual research is going on. Okay, so now that you know how we grow bacteria, we're going to talk about the interesting aspects of bacterial genomes and their genetic exchange. One thing I want to point out about the right side, all those little dots on that bacterial plate, each one of those represents a bacterial colony, and each colony in a bacterial culture like this is a visible population of bacteria, meaning it's not a single bacterium sitting there, it's thousands or tens of thousands of bacteria, and each of those colonies on the plate is the descendant of a single bacterium. So what I might do is take some water from the water fountain, right? Take 1 drop of water from the water fountain and spread it across the top of this solid media and let it sit for a night or two. And if I come back and somebody's been mouthing the mouthpiece on the water fountain and gotten their bacteria on it, you could see growth like this. And what that would represent is when I spread that drop of water around on the plate, every little bacterium that kind of found a place to settle and grew overnight on my plate, it grew not by that individual cell getting larger, but that cell divided and divided and divided and divided, right? So you have thousands of descendants of that one cell. Okay, so in growing bacterial plates, we can culture bacteria in this way, and we can culture lots of different individuals. And this will become important again close to the end of this lecture as we talk about some of the ways of screening bacteria for genetic variation. All right, so the 3 primary things that we're going to touch on with respect to the transfer of genetic information within bacteria are conjugation, transformation, and transduction. These are briefly introduced on page 146 in your text, but we're going to go through each of them, some experiments that have been done, and explain how these bacteria can exchange information, take up new information, give information to other, when I say information, I mean genetic material, give information to other individuals, and why this is important. So conjugation is the first of these mechanisms that we're going to discuss. And in conjugation, one bacterial cell is passing genetic information to another. Okay? And if it simply passes it over, that's one thing, but the important part is that the recipient, the cell that receives genetic information from another, must genetically recombine its own genome with that new material. Okay, So the recipient bacterium takes in this new genetic information and sticks it into its own genome. Sometimes it might be at the expense of an existing gene, it might replace it, or it could be added to the existing genome. And so, one thing I want you to keep in mind is that the movement of this genetic information among bacteria can be categorized or classified as horizontal or vertical. In vertical transmission, what we have is a single species in which different individuals are exchanging genetic material. Okay? So bacteria do not reproduce sexually like humans do, right? So when you produce offspring, it's with a mate, and those offspring are basically the fusion of your genomes, portions of your genomes, okay? Bacteria don't reproduce sexually. So in order for there to be genetic diversity within the population, for them all not to be identical clones of one another, they have to mutate, which we know they do, but they also are able to exchange genetic material with one another within a species through vertical gene transfer. Now, horizontal gene transfer is a completely different animal, and this is one in which two different species exchange genetic material. Now, it might seem that this is something that we probably wouldn't care about or think about. Why does it matter if bacteria exchange genetic material within or among species? But keep in mind that many of the diseases that affect humans and other species, the maladies, are caused by bacterial infections. For us, we treat most of those with antibiotics, right? But you are probably aware of things that you've probably heard the term antibiotic resistance. There are strains of bacteria that are resistant to antibiotics. And the way in which that can spread among a species or among different species is through horizontal or vertical gene transfer. Okay, so if you've worked in a hospital setting, you might be aware of a bacterial infection that's quite common and quite feared in hospital settings, and that's MRSA, and that is, or MRSA, that is a strain of bacteria that is resistant to commonly used antibiotics, and it's very difficult to get rid of this infection. Well, think about how are those bacteria resistant to that antibiotic? Okay, there's been a mutation to some gene, they are no longer susceptible to it, but think now about the possibility of horizontal gene transfer. What if other species of bacteria are able to gain that genetic-based resistance to antibiotics and therefore resist our attempts to eliminate them from the body when a person is infected or sick? Okay, so that's just giving you an example, a human health example of why these horizontal and vertical gene transfer mechanisms are important. But let's dig into now how these actually happen. So here's an experiment that's illustrated in figure 8.2 of your text, in which it shows two different strains of bacteria that are being grown in liquid culture. Okay, so they're in these flasks. We have strain A on the top left, strain B on the top right, and underneath the strains you will see these three-letter abbreviations. Okay, these indicate 5 important organic compounds that these bacteria need to survive. Okay, and in strain A, what those 5 different symbols indicate is that strain A is capable of producing the last 3, THR, LEU, and THI. They have the wild type. Remember that plus sign. So they're capable of producing those 3 compounds, but they're incapable of producing MET and bio. All right. So what this means is, to grow strain A, you have to put it in a liquid culture that contains all of the necessary things that it cannot produce on its own. Okay, and so you see in between these two flasks, it notes that these are auxotrophic strains. Remember, that means they can't produce everything they need, and they are grown in complete medium, not minimal medium, complete medium. Complete medium gives them everything they could possibly need, things, including the things that they should be able to make on their own. So if you supplement their media with all of these things that they can't make, then strain A will grow. You'll notice that strain B is able to produce MET and bio, but incapable of producing the last three compounds that strain A is able to produce. So they are complementary in terms of what they can and cannot produce, but neither of them can grow in minimal media. They can both grow in complete media because everything they can't make is provided in the media. Now what's interesting is we've got these two controls, so strain A and strain B, follow the arrow straight down from them. If you take some of that liquid and you spread it out on a plate of minimal media, okay, this is a plate that has media in it that is not supplemented with any of these compounds that the bacteria should be able to produce on their own. And you'll notice that in the controls, neither strain A nor strain B are capable of growing on this minimal media plate. But what is really interesting is when you mix A and B into a single flask, you put some of each in there and let them grow overnight. And the next morning, you take some of that liquid culture, in which there are certainly many more bacteria than there were yesterday. You plate them on minimal media, and what's going to happen is you are likely to observe a few colonies of bacteria grow on this media. Now remember, each of these colonies are essentially the descendants of a single bacterium, okay? So here we see 3 colonies grew. That means of all the bacteria you put on, let's say you put 20 microliters of this fluid culture onto this plate, presumably thousands and thousands, tens of thousands of individual bacteria, and spread them around on this plate of minimal media. The vast majority of them did not survive. They did not grow into colonies, but 3 of them did. It's a very low ratio. It's a very low proportion of the entire sample that was put on that plate. But what this means is, because they're growing on minimal media, is that those 3 individual bacteria are able to produce on their own all 5 of those compounds, okay? Which is shocking because neither strain A nor strain B were able to do that, okay? So how are any of these bacteria able to do it? Because we know that these bacteria are not sexually producing offspring, right? They are not going to be heterozygous and therefore able to produce each of these compounds. And so the question was, are these bacteria simply releasing their DNA into the media so that it can be taken up by other individuals and they then become prototrophs? So the next experiment tested that hypothesis. Are they just releasing DNA into the media? And the answer turns out to be no. And an elegant experiment in which this was tested used this U-shaped tube where at the base of the U there's a filter that will allow the liquid to pass back and forth, but it does not allow the bacteria themselves to move back and forth. And so it's a pretty large holed filter. Everything but the whole bacteria themselves can move through it. And so if they're releasing, let's say, DNA into the media, you can see that at the top of the tube, pressure and suction are alternately applied. So it kind of pushes liquid back and forth across that membrane, but the cells are kept separated. Strain A on the left, strain B on the right. And so if they're just releasing this genetic material into the media, and some individuals are absorbing it, taking it in, recombining themselves and becoming prototrophs, we ought to see that same thing happen here. But it turns out that that does not happen, okay? And what this tells us is that the cells must come into contact with one another. They can't be separated from one another. There needs to be physical contact between the cells for there to be this spontaneous generation of prototrophs. And so let's talk about how conjugation works. Okay. Now, I'm going to start out by talking about these notations that I didn't mention before. You'll see that before strain A, there's an F plus, F with a positive superscript, and with strain B, there's an F with a negative superscript. These would be referred to as F positive and F negative cells. And what this refers to is whether or not the cells possess what is called an F factor. So let's take a peek at this next slide, and I first just want you to think about the cells and their genetic material. So here we have two cells, okay? An F positive cell and an F negative cell. The difference between them is this blue circle, okay? The yellow circles are here and meant to indicate the primary or the main bacterial chromosome. Each of these cells, the F positive and the F negative, has a bacterial chromosome, okay? And these are shown as two concentric circles simply to indicate that this is double-stranded DNA, okay? Now you'll notice the F positive cell has this additional blue chromosome, okay? This small blue double-stranded DNA, what is called F factor. And you'll note that in red on the left, this cell has been indicated as being the donor, okay? Now cells can come together like this and conjugate. That is that physical connection between the two cells. You see that tube, okay? So conjugation, you've probably heard terms like a conjugal visit for somebody that's in prison. So if you've watched Space Force, right? He went for a conjugal visit to the prison with his wife. These two bacteria here are having a conjugal visit. Okay, this conjugation is made possible because one of the cells has an F factor. If there's no F factor, this isn't going to happen. Okay, so that's the first thing to understand. Once these bacteria form this conjugation tube, this connection, something happens with the F factor. And remember that F factor is double-stranded DNA. Well, what happens after the conjugation tube is formed is one of those DNA strands of the F factor is nicked, and that strand begins being pulled into the F negative cell or the recipient cell, okay? Now, once bacteria recognize the presence of single-stranded DNA within their cells, whether it's the upper F positive cell whose F factor has now become single-stranded because the other strand is being pulled into the F negative cell, or if it's the F negative cell in which a new strand of F factor is being pulled into. And so both of these cells have some single-stranded F factor DNA, and that's not a stable state for DNA. So when this is recognized by the bacterial cells, they begin to synthesize a complementary strand to this single strand of F factor DNA. So they are both making a complementary strand of F factor DNA as this process goes on. So you can see here the new DNA that's being synthesized, and what it does is it makes each of their F factors double-stranded. After this process is complete, after a whole F factor strand is pulled into the new cell, the F negative cell on the bottom, and both have made complementary copies of their F factor single strands of DNA, they will both end up with double-stranded F factors. They will close the conjugation tube. They are again two separate cells, but they are now both F positive. Okay? So this is a way in which genetic material is passed from one bacterium to another through conjugation and the movement of the F factor from one cell to another. Now notice that the donor cell doesn't lose its F factor, okay? They simply both become F positive cells. Now, in this conjugation, only the F factor is moving. But what was interesting when scientists realized how conjugation worked, they started to notice that on occasion, some strains that they had in the lab had a high frequency of recombination. Okay? So it was things were changing in the recipient cells, not just the presence or absence of an F factor. Okay? So these cells were classified as high frequency recombinant cells, so HFR, and these strains were used to map bacterial chromosomes to get an idea of what else is on the bacterial chromosome and what is the order of all of these things on bacterial chromosomes. Now, you'll see why that becomes important later when we talk about transformation of bacteria. But for our purposes, I want you to understand that that main yellow chromosome we saw on the previous slides, that contains nearly all of the genes for the bacterium. Now, if two different bacteria contain different alleles, you can detect recombination. You can detect new combinations of alleles within individual bacteria. And this was observed to be the result of recombination taking place when these HFR strains interact with one another. So let's look at how that happens. Now, what we start out with at the beginning here is another F positive cell, right? A normal bacterial chromosome with 5 genes on it, A, B, C, D, E, okay? And in this, we've got all capital letters. Let's just call those dominant alleles for genes A, B, C, D, and E, okay? Now remember, bacteria are not diploid like you and I. They have a single copy of each gene, okay? And so they can't be heterozygous, they can't be homozygous, they just have one copy of everything. In this cell, the F factor, if you look at the second cell down from the top, the F factor integrates with the bacterial chromosome and becomes part of it. Okay, so rather than being its own separate circular double-stranded DNA molecule, it becomes part of the larger bacterial chromosome, which you see still has those 5 genes, but also has the F factor in it. Okay. Now, one thing that's important in these HFR cells is the orientation of that F factor. Think of taking a double-stranded DNA circle and making it linear. You could stick it into that orange bacterial chromosome one way or flip it around and put it in the other way. It doesn't really matter to the bacterial chromosome, okay? But where it matters is when conjugation takes place, as you see on the bottom right, and that O representing the origin of where the F factor would normally be cut and start being pulled into the recipient cell, okay? It is that the position of that origin that is important when these two cells begin conjugating. So on the bottom right, you will see that this HFR cell, which is itself still F positive, right? It still has an F factor in there. That F factor is just stuck into the bacterial chromosome. This HFR cell can form or conjugate, can form a conjugation tube with an F negative cell. And notice that in this case, the F negative cell has different alleles, the recessive alleles for A, B, C, D, and E. So when this conjugation happens, we see that what begins to be pulled into the recipient cell, in this case, because of the location of the origin on the F factor that it was integrated into the HFR cell's chromosome, what gets pulled in starts with the big A and keeps pulling and might take the B with it, could even take the C. It just depends on how long these cells are conjugated, okay? And it turns out that these cells, in this particular illustrated instance, are connected long enough for all of gene A, all of gene B, and not much more to make it through. The cells, they can't really maintain this connection in perpetuity, right? They're getting bounced around by their neighbors that might be moving around in the fluid. Maybe, a lab technician picks up the vial, something happens, okay? So they don't necessarily remain conjugated forever. In this case, conjugation ends, and what has been pulled over is not even the F factor, okay? But what got pulled over was a chunk of the chromosome that contained the dominant alleles for gene A and gene B, okay? And so the X conjugates consist of a cell that has the normal, what we started with, the HFR cell, A, B, C, D, E, dominant alleles, and the F factor stuck in there. But the other one, the recipient, received a portion of the bacterial chromosome that contains dominant B and A. It was made double-stranded after it was brought in. And now that cell has 2 copies of gene B, 2 copies of gene A, all right? So they are actually diploid for those 2 genes right now, which is kind of an awkward scenario, okay? So they have both copies, 2 copies of that gene, both alleles of that gene, and this can persist for a while. Sometimes the bacterium might just dissolve that random chunk of DNA that's in there. Sometimes they will stick it into their chromosome and get rid of their recessive A and B, okay? But this is an instance in which the conjugation has resulted in the change in the genotype of specific genes that are present on the bacterial chromosome. Now, this F factor that has been incorporated into the bacterial chromosome in an HFR cell, the bacterium can recognize this and cut it out, excise it. And you can see it in this second cell from the top that the bacterial chromosome's kind of been twisted into an infinity symbol, and this is in an attempt to cut out the F factor. And in doing so, what the cell ends up doing in this illustration is cutting out not only the F factor, but also sending away with it genes A and E, okay? So this cell, it's not F positive, it's not F negative, it's not HFR. It is what we call an F prime cell. Now, this excision of the F factor and creation of an F prime cell due to the incorporation or inclusion of some of these chromosomal genes into the F factor creates another interesting opportunity for the interaction or conjugation of bacterial cells. So here we have on the top right an F prime cell that is going to interact with an F negative cell. Okay, they have different alleles for all 5 of the bacterial chromosomal genes. And when they form this conjugation tube and the F factor moves from one cell to the other, what we end up with is on the top left, you will see a merazygote. Okay, this cell, unlike the one on our previous slide, where they had 2 copies of gene A and B, but one of them was just a linear piece of DNA floating around and probably not terribly stable, could be problematic. This merazygote, this F prime cell, is stable. It has its own F factor, it has its bacterial chromosome with all 5 genes on it, but for genes A and gene E, it is diploid for those and will remain diploid. Okay, that's not a problem for the cell. So every time this F prime cell interacts with or conjugates with an F negative cell, it's going to give them the F factor and the dominant allele for gene A and E. Okay, so some really interesting mechanisms of genetic exchange within bacteria through this process of conjugation. Now, I want to introduce you at this point to a term that you will see used if you take micro, and that is plasmid. Okay, plasmids are generally what we use to classify circular chromosomes that occur frequently in bacteria. Okay, this circular double-stranded DNA is something that is highly useful in genetic laboratories, And these plasmids may contain a number of different genes. Okay, so you can see here there's a plasmid with a number of different genes indicated along it. Okay, you don't need to remember any of these. Okay, but what's important about this plasmid that is illustrated is that on this plasmid are a number of different genes that confer resistance to antibiotics. Okay, so if this plasmid were stuck into a bacterium, it would give that bacterium resistance to certain antibiotics. And that seems like a terrible idea. Why would we do that? Okay, well, it becomes a very important screening tool for genetic laboratories, because sometimes we want to put things in bacteria and we need to know if the things we tried to put in the bacteria are actually in there. And so we will grow the bacteria in the presence of antibiotics. And if the bacteria grow, we know they got the plasmid that we tried to put in there. If they don't grow, if they die, then they didn't receive what we tried to put in them. Now, to be clear, we're not using bacteria that are infectious to humans. It's generally not a situation that's dangerous to the health of the researchers doing the work, but it is an incredibly powerful screening tool. And I'm going to give you some examples in a bit of how we can use screening to determine whether or not bacteria have received the things we've tried to put in them and why that might be important. But first, I'm going to introduce you to the principle of what we call transformation. So unlike conjugation, where two bacterial cells come together and exchange genetic material through this conjugation tube, in transformation, we use what is called a competent bacterium. So it is capable of receiving genetic material that we want to put into it. And in introducing this genetic material to the broth or the mix that contains this bacteria, we are able to hopefully get these bacteria to incorporate this DNA, to bring it in, to let it into their cells. So most of the time, if we are exposed to DNA, right, if I'm in the lab and I spill a tube of snake DNA onto my arm, my cells are not going to take up that snake DNA and me start expressing snake genes. I'm not going to grow scales on my arm, okay? Because my cells are not competent in that way. They are not capable of taking in DNA and assimilating it into my genome in those cells. But in transformation, we use bacteria that are receptive, they have these receptor sites, to taking in DNA. And this DNA acceptance or absorbance, if you will, can be initiated by either some temperature shock or an electrical shock. So we kind of shock the cells, and you can kind of think of this as the cells gasping or inhaling, and they take in DNA that we've put all around them. If I want them to take in a particular gene, I put millions of copies of that gene into that culture and then shock the bacteria so that they suck it up. Okay? We'll talk about how we're able to make this transforming DNA, this double-stranded DNA that we want to take these bacteria to take in. It's a relatively straightforward and simple process that can be done in lots of commercial labs. So if I want to order a gazillion copies of a gene, I just put the order in online, pay $20, hit submit, and the lab will send it to me within a few days. Okay? So in this process of transformation, we shock these competent bacteria to initiate the entry of this DNA that we've put in there. And once this DNA enters the cell, this double-stranded DNA will generally denature, so separate into two single strands. And one of those, the transforming strand, can bind to regions of the bacterial chromosome and swap themselves in. So through complicated cellular machinery that is more detailed than we're going to get into, this heteroduplex can be formed where one of the original bacterial strands is cut and thrown out. The new transforming strand is stuck in there, and the bacterial cell can then even take out the orange strand that still remains there next to the heteroduplex and fill that back in with DNA, single-stranded DNA that is complementary to this blue transforming strand. And so what we end up with is what you see on the top left. This transformed cell, so it's a bit cut off where it says that, and below it the untransformed cell. And the transformed cell is one in which this transforming DNA has been assimilated, and the heteroduplex was formed, and new DNA was brought in to replace the original yellow strand there. So we have a double-stranded blue section of that bacterial chromosome. In an untransformed cell, this doesn't happen, okay? You can see that in this cell, in step 4, where this heteroduplex exists, the other way that we can end up with double-stranded blue DNA in a transformed cell is if that step 4 cell sticks around and survives in this odd heteroduplex state, that bacterium is going to divide pretty soon, right? Within minutes to hours. It's going to split into 2. When it does that, we know that DNA replicates just like it does in mitosis and meiosis. Okay, so these cells have to undergo DNA replication so that when one cell becomes 2, each of those bacterial cells has a double-stranded chromosome. Well, think about if you take one of each of these strands and replicate them, you're going to end up with a transformed cell that has a double-stranded blue bit, and the cell that gets the original orange copy that was then duplicated will appear as this untransformed cell. Okay, so after one round of DNA replication, you end up with a transformed cell that has double-stranded blue. Okay, and we are going to get into a great bit of detail about how DNA replication occurs, but simply know that when one cell goes to 2, as we've learned in mitosis, the DNA has to be replicated so that it can be distributed to each of the daughter cells, each of the 2 cells that results from the split. And in this case, it leads to a transformed cell that is homozygous for this transforming strand, if you will, or has only, not homozygous, sorry, it has double-stranded DNA reflecting the incorporation of this transforming strand. Okay, so what do we do with this transformation? Why is it important, and how can we use it? Well, one of the great examples, one of the earliest examples of how bacterial transformation has been of benefit to humanity is the production of insulin. Okay, so this is something that, in folks with diabetes, they are incapable of producing this, and so they have to supplement by daily or periodic injections of insulin. So how do we get insulin? Well, before we were able to do this, before we mastered transformation, insulin had to be gathered from slaughterhouses. So we had to go collect insulin by basically milking the organs of other mammals that we slaughtered, like cows, and try to extract it. That's a very difficult process and time-consuming and doesn't yield a great deal. But with transformation, what we were able to do is take the human gene for insulin and stick it into a plasmid, okay, and then put that plasmid into a bacterial cell. So you can see here this plasmid, it's got these different colored regions, and you can see that the plasmid was cut open, okay? It went from being a circular double-stranded DNA molecule to being a linear, even though it's still in almost a semicircle shape. It looks more like a C. But it was cut, the double-stranded DNA was cut in a way that opened it up and made it possible for us to get this insulin gene into that bacterial plasmid. Okay, so the ends of the blue insulin gene and the white plasmid here are glued to one another using an enzyme called DNA ligase, and we'll talk a good bit more about ligase when we do DNA replication. But this DNA ligase can be used to glue insulin or stick it into this plasmid. We end up with a recombinant plasmid, and we put that plasmid into a bacterium, and that bacterium can now express this gene. It can now produce human insulin. And it's very easy to extract insulin from a vial or a flask or a beaker of bacteria. It's slapped full of insulin. Okay, and so we just extract it from there, refine it, and then distribute it for use by humans with diabetes. Now, this type of genetic engineering, if you will, is not 100% effective. Okay, we're going to make a lot of these recombinant plasmids. We are going to try to put them into a lot of different bacteria, but not all of them are going to receive the plasmid. Not all of them are going to take it in and begin expressing this human insulin gene. So what we do is we utilize our understanding of the genome of these bacteria in a way that allows us to differentiate between those that do and do not have this plasmid with the insulin gene in it. Okay, so while it's not 100% successful, we can screen out and get rid of all of those bacteria that don't have what we tried to put in them. So here's an example in which we have a plasmid on the left, and we're trying to put this foreign DNA in there, okay? This red bit of double-stranded human DNA, just imagine it's the insulin gene, okay? And what we do is when we cut the plasmid that's on the left with a restriction enzyme that was mentioned on the previous slide, we cut that double-stranded plasmid, and it cuts only in a specific place. We know it is going to cut right in the middle of this beta-galactosidase coding gene. Now, you don't need to know what beta-galactosidase does, but what happens is if we cut it there, and if we are successful in getting this foreign DNA stuck to that spot, look at the resulting second plasmid there. You see that there's a double-stranded red bit stuck right in the middle of that beta-galactosidase coding gene. So what this means is we've completely interrupted that beta-gal gene. Okay? And so if we put these plasmids into bacteria, okay, we try to make a bunch of these recombinant plasmids. We try to put all of those into thousands of bacteria in a flask, and then we grow them on plates or in culture. And the hope is, if this is an insulin gene, that all the bacteria get the insulin gene, all of the bacteria produce insulin, and we have high efficiency production of insulin. But there are three possibilities for the outcome within our flask. If you look at the top option here, some of these bacteria will receive a plasmid. They'll take it in. Okay, the bacterial genome is present in all of them, but some of the bacteria take in a plasmid. But the plasmid that they take in might not have had that foreign DNA incorporated into it. And if it didn't, then it has an intact beta-galactosidase gene. And what this does is it makes the bacteria appear blue. So any of those bacteria that grow on our plate and have a blue color, we know that they have the plasmid, okay? But we also know that they did not take up the insulin gene that we tried to put in there. Now, the middle option, this bacterial cell, it has its bacterial chromosome. It does not have a plasmid, okay? Now, the one thing I hadn't yet mentioned is this other gene that's indicated on the far left plasmid, that lighter blue gene that confers resistance to tetracycline. If the cells don't take up any plasmid at all, neither the one with or without the beta-gal gene disruption, if they don't take up a plasmid and we put them on these bacterial plates, we have put the antibiotic tetracycline. If they don't take up any plasmid, they don't grow at all. Okay? And the third possibility is, at the bottom, that the bacteria take up a plasmid that does have this foreign DNA, this insulin gene, inserted in the middle of the beta-gal gene. And what this means is these bacteria are resistant to tetracycline, but they do not produce this blue coloration. And so these three options all manifest themselves in these plates. And you can see here what this looks like in real life. If we plate all of these bacteria that we tried to transform, some of them are going to be blue. That means they took up the plasmid. The plasmid did not have the insulin gene inserted into that beta-gal gene. It wasn't disrupted, so they still produce blue coloration. The white colonies, those did receive a plasmid. That plasmid does have the insulin gene in it. And so if I want to grow and produce lots of insulin-producing bacteria, all I need to do is take one of these, touch it with a toothpick, drop that toothpick into some liquid culture, and I can grow gallons and gallons of liquid culture of bacteria that have the human insulin gene in them, and they produce that protein that is needed by diabetics. Now, what you don't see here are all the colonies that would have resulted if there were no antibiotic on this plate. All the bacteria that didn't take up any plasmid are dead. Okay, they did not grow at all. So that's transformation. Okay. Now, to talk about the next mechanism of genetic exchange, transduction, I need to introduce you to the fact that bacteria are just as susceptible to viruses as we are. And the viruses that infect bacteria are called bacteriophages, okay? And they're quite simple in that they have this protein body, which in the image below is pretty much everything that looks like the gray lunar landing module. And then they have DNA or RNA, depends on the virus. But the head of these bacteriophages is packed with this genetic material, indicated here by the purple squiggly line, okay? And so what these viruses do is they will attack a cell, just like you see here in the top. So this bacterial cell, a bacteriophage lands on it, if you will, and the phage is absorbed into the host cell. And what that means is it basically inserts its genetic material into the cell. And from this point on, that cell is hijacked completely. Whatever it was doing, it no longer does. Whatever its intents and purposes were, they are now under the control of the viral DNA, okay? And the first thing that viral DNA does is chop up that host chromosome. No longer need that. What the viral DNA does is, after it destroys all that bacterial DNA, is it uses all of the raw materials present in that cell to make more viruses. This is exactly what happens in your cells when you are infected with a virus, okay? Your cells take in viral DNA, the viral DNA takes over control of those cells, and it makes more and more viruses. And then, as you can see in step 5, the cell bursts and releases many, many mature viruses that will infect your other cells, but also be released so that presumably you infect other individuals and the whole process starts all over again. This is obviously very relevant today, and we're all thinking about viruses more often than we ever have. But in bacteria, this is the way in which bacteriophages infect bacteria, okay? Now, what transduction is it's a relatively uncommon, or far less common than this, process whereby this infection of a bacterium by a phage takes place. But what we haven't talked about so far is that in this, it's possible for the little tiny particles of the bacterial chromosome that were chopped up When the phage makes more phage pieces and puts them all together in step 4, sometimes when it is packing the genetic material into the head of the virus, it doesn't put in there viral DNA, it puts in there one of those chunks of bacterial chromosome that were still floating around. Okay? And what might happen is in step 5 is one of those viruses, one of those bacteriophage that's released when the cell lyses. So the lysis is the breaking of the cell and release of all these viruses that were created. After lysis, some of the phage that go infect other cells carry with them not viral DNA, but they carry with them a little bit of bacterial DNA. And if they infect another species of bacteria or an individual of the same species, so whether it's horizontal or vertical, so if it's a vertical that has a different allele, they could bring different alleles to the same species. Or this could be a way in which genetic material jumps from one species of bacterium to another if it's carried by a virus, because these chunks of bacterial DNA can be incorporated into the recipient's chromosome. So you can see here on the left, this blue chromosome is going to receive a double-stranded chunk of orange chromosome, potentially recombining and introducing new genetic variation, maybe even brand new genes to a different species or a different individual of bacteria. So I want you to realize that this is a very real way in which genetic exchange between bacteria can occur. And this is potentially quite frightening if you think about antibiotic resistance in one species of bacteria. This is a very easy way that it could jump from one species to another. Okay? If the gene that allows species A to resist an antibiotic is wrapped up in a virus and carried to species B, all of a sudden now we have species B also resistant to that same antibiotic. Okay, so when we think of phage, there are a couple of terms that get a bit confusing, so I've listed them all here. I encourage you to review the text so that you are very familiar with the usage of these different terms. The first of which being lysogeny. So this is a scenario whereby the cell doesn't exactly undergo the process that I illustrated, where viral DNA comes in, takes over, makes more, and blows up the cell, releasing a bunch of viruses. A lysogenic virus is one that is repressed, so it doesn't come in and just take over everything. It basically goes in there, incorporates itself into the bacterial chromosome, and hangs out. Okay? It might be a bit of a sleeping giant, but it is hanging out there for the moment, not doing anything. A prophage refers to that viral DNA that integrates into the chromosome. Phages that are classified as temperate phages rather than virulent, which we'll get to in a second. Temperate phages are viruses that either can lyse the cell, they come in, take over, make more, blow it up. So temperate viruses are those that can go in and do that, they lyse the cell immediately, or they can opt to take route B and act as a prophage, which is described above. Okay? Now, virulent viruses, they have only one course of action, and that is come in, take over, make more, blow it up. Okay? And then a lysogenic bacterium, that is one term that describes a bacteria that has a prophage integrated, so the viral DNA incorporated into their chromosome. Okay, so just be comfortable and familiar with these terms. All right, now the last thing I want to touch on here is the growing recognition of the importance of bacteria in all aspects of biology, evolution, and human health in particular. Okay, this is becoming increasingly apparent and important as we consider, the broad range of health conditions that affect humans, but also of great interest to evolutionary biologists as they study how the diversity of life has evolved through the interaction of species. And I don't just mean the interaction of cheetahs and gazelles, but also the bacteria that live in and on you. So what you might not realize is that your microbiome, which is the term we use to refer to all of the bacteria that are associated with you, there are more bacterial cells in and on your body than there are human cells, and by a factor of 10. Okay? So you have lots of human cells. There are about 10 trillion of you. But there are 10 times that number of bacteria just in your gut. All right? And they are hugely important to your health and well-being. It's not something that you need to hand sanitize away or go drink bleach to kill all of these things because you don't want bacteria inside you. If you don't have them, you don't exist. Okay? And if you don't have the right ones, then it can affect your health very dramatically. Okay? So one example of the importance and characteristic nature of your microbiome, just look at the region and the regions of the body and the associated bacteria. Okay? Each of these portions of your body has loads of bacteria on it. Don't go scrub it off. You need it there. If you don't have the right bacteria in the right place, you develop skin conditions, you develop rashes, you can develop serious problems. And each of these regions has a very characteristic type of bacteria. If you see how these pie charts are colored, you can see that the area of your head has very different bacteria than do your arms or your chest. Okay? These are very high level classifications of microbial organisms. And so the diversity, it's not just like you have very closely related species in different parts of your body. It's that the things under your nose are so different from the things on your palm or in your elbow crease. Okay? So you have a very characteristic biota, and if this gets disrupted, problems arise. All right? I'm just going to give you one example of studies that have been done on the influence of the bacteria that you have in your gut and how that affects one aspect of the phenotype, and that is obesity. So this is a study that was published. Remarkable findings. Okay, what they did is they took identical twins, but these twins, for some reason, were very different in their obesity, right? One slim twin or lean, and then one obese twin. What they did is they took fecal material or bacterial samples from the gut of these human twins, and they gave those, by contaminating the food, of mice that basically had virgin guts. Okay? You can raise mice to have sterile, like they have no gut bacteria at all, and they introduced to those mice the bacteria present in each of the twins and fed them the exact same diet, those mice, for a period of time. And what they found is, even though they're eating the exact same thing in the exact same amount, those mice that were exposed to the obese twins bacterial culture became obese. And the mice that consumed or were contaminated with the lean twins microbiota remained lean. Okay? So it's not the food, it's not how much they eat. It had everything to do with how the bacteria in their gut were processing the food. Okay? Remarkable. And then what they did is they put the mice together. They took those mice, one became obese, one remained lean, and they housed them together and let them live together and eat together. And what they found was that not only did their bacteria become more similar, they found that depending on what they fed them, the obese mouse became lean and the lean mouse remained lean. Okay? So this lean, it's not healthy for us to be obese. Evolutionarily, we're not intended to be obese. We should not be associated with bacteria in our guts that make us obese. And so when the bodies have the availability and the option to take in bacteria that help them better assimilate and digest food, that is the population of bacteria that will establish itself in the gut. If you look here, look at columns 1, 2, 3, and 4 in both of these, the left and the right side. This is a very complicated figure. But on the left, we're looking at the lean mice. On the right, we're looking at the obese mice. See up top where it's LN versus OB, lean and obese. Before they were housed together, look at how these cells are colored for 1, 2, 3, and 4. Okay? The lean mice have lots of red rectangles, okay? And very few blue rectangles in columns 1, 2, 3, and 4. And what that means, if you look to the right, it means that the lean mice had lots of these bacteria sort of in the top half of this chart. And the obese mice, they have lots of blue, right? And so they have lots of these bacteria that are in the bottom half of this table. Once you put them together, look what happens, okay? You put them together, and the obese mice, they get more and more of the red squares. And the lean mice start picking up more and more of these blue rectangles, okay? So interacting with one another changes their microbiome, okay? Their diet, this is not to say you could have a healthy gut microbiome and just eat, Little Debbie snack cakes 100% of the time and be skinny, okay? But this does give us some clue as to why some individuals possibly have what appears to be a healthy diet and exercise regimen, but they have troubles dealing with obesity, whereas others do not. And it's these types of health issues and microbial imbalances appear to be linked to a number of health conditions like obesity, like Crohn's disease, where people have difficulty consuming certain things, IBS, where people have bowel troubles. And that's why in a number of countries, so you can go to Australia and get what is called a fecal transplant surgery. Well, that's exactly what it is. You basically have a chunk of poop from a healthy person's gut put into your gut. And what you're doing is introducing into your gut the right mix and balance and diversity of bacteria that can properly digest and deal with the foods that you're eating. So this is just to introduce to you the importance of bacteria and give you some clue as to the interaction of bacteria, their genetics, and humans and our genetics, because it's not just the presence or absence of bacteria, it's also the interaction of those bacterial genomes and the things that are produced by those genomes with our bodies and our genomes. Okay, so that is all for this chapter.